Hello, everyone. Welcome to Brainmap. This seminar series is co-sponsored by P40 funded Center for Mesoscale Mapping, housed in the Martinos Center. It's my great pleasure today to introduce our speaker, Dr. Nathan Rose. Nate completed his PhD in Behavior, Brain, and Cognition program in Washington University, St. Louis, and then postdoctoral fellowships at the University of Toronto and the Rothman Research Institute at the Bycrest Center for Geriatric Care and Psychiatry Department at the University of Wisconsin-Madison. He joined the faculty at Notre in 2016, where he directs the Cognitive Science, Memory and Aging Lab and co-directs the ND Virtual Reality Lab. Dr. Rose studies the neurocognitive processes that support working memory, long-term memory, and prospective memory in healthy young adults and healthy other adults, as well as in patients in Alzheimer's disease, Parkinson's disease, and or amnesia. His research uses neuroimaging and neurostimulation technologies, behavioral assessment, to test and inform the theories of memory and aging. In addition to studying basic memory processes, his research also assesses how cognitive theories can be applied to understanding memory performance in real world and how cognitive training and brain stimulation techniques can be utilized to improve memory maintenance. I would like to remind our audience that address their questions at the end of the talk, either in the Q&A box or raising their hands for virtual audience, and by raising their hands here in the in-person audience. Also reminding that in-person audience has priority for the questions. So please enjoy the talk. Dr. Rose, Nathan, thank you for coming, and the stage is yours.
All right, thank you, Isha. And thank you all so much for having me. I'm thrilled to be here to kick off the Brainmap Seminar Series for the year. Happy New Year, everyone. And I'm going to go ahead and get started. This talk, using non-invasive neuroimaging and neurostimulation to measure and modulate memories in humans. This is not a methods talk, despite the way it may sound. I want to talk about how these techniques are allowing us to address theoretical questions about how they can help us to constrain and advance theories and models of memory and aging from both basic and applied perspectives. And I'm really interested in using in learning how we can use these technologies going forward. So I'm really excited to hear your thoughts and perspectives. Okay. So the talk, hopefully I'll be able to get through two parts. The first is the main part about revealing how working memory works, using fMRI-guided TMS EEG as a neurophysiological probe to assess the existence of these latent or activity-silent working memories, and how we're using it to advance this synaptic theory of working memory, as well as a long-term memory account of latent working memory. Long-term memory account using assessments of subsequent long-term memory tests, as well as assessments in healthy older adults with Alzheimer's disease biomarkers. And then the second part, hopefully I'll get to just briefly touch on a systematic review and meta-analysis of the ways in which TMS may be used to enhance or affect long-term memory, and provide an example of how we have recently used it to modulate emotional memories. Okay, so part one is about how the mind and brain holds information in working memory, in scare quotes, and in particular, how we get information that was just in the focus of attention back into working memory after attention shifts away from actively maintaining the information. So everybody will be familiar with this kind of a situation. Consider a situation where you meet my senior thesis student, Savannah, on a plane, and she wants to go to grad school. So you're chatting with her about that. And then in walks Samuel L. Jackson. And you're like, oh my gosh, what is Samuel L. doing on this plane? You're distracted and you're like, oh, I should ask him a question. What is he doing on this plane? Maybe there are snakes on this plane. Good, a few people still got that laugh. I hope that never gets old. And then you realize, oh, how rude you're being to this nice person you're just chatting with. And so your attention shifts back to her and say, oh, what was that, Samantha? Oops, faux pas, her name's Savannah. How rude. And the point is that according to models of short-term or working memory, when you are focusing on Savannah the first time, this is the item that's actively maintained in working memory. And then when your attention shifted to Samuel L., this item is no longer actively maintained in working memory. And the real question is, how do you get this information back when you need it later on for goal-relevant cognition? This is a situation that everybody has experiences with. And the question is this item still in working memory, or has it really been dropped from working memory so that you need to reactivate it from long-term memory using episodic retrieval processes, and so that you can use it going forward? This is the question that we're really focused on in this situation, and we use it in a similar kind of a two-item working memory, really simple working memory task with these retro cues to tell the participant which item they're going to be tested on. So that causes them to shift their attention to the cued item, and then later on shift their attention between items that are maintained. And brain decoding methods with neuroimaging data can help us track the neural representation and activation of working memory items as they transition between attended and unattended states. And the way we and others have done this is by presenting two items, and we can decode both items when they are initially presented and retained on both these cue switch and cue state trials. And then when the red one is cued, decoding remains elevated for the cued item, but the blue one drops all the way back down to baseline levels of representation, as if it's no longer in working memory. But we think the unattended item is in working memory because we can cue the participant to switch attention back to focusing on this initially unattended item, and participants can rapidly and accurately do so, and we see a corresponding crossover in the pattern of neural representation. And this finding has been replicated several times now in fMRI, EEG, even item level decoding with fMRI, and most recently, even when the items remain on the screen in a attention selection or change detection task, at least with decoding the neural signals from ventral occipital temporal cortex for the contralateral item. So this evidence of so-called activity silent retention in working memory is really quite different from the long-held assumption that information must be retained in working memory with an active maintenance mechanism. So the question, how are these unattended items retained if not by an active neural trace? One theory that can potentially account for this phenomenon is a computational theory by Sodek's group, which essentially posits that a feed-forward sweep of structured activity in a network, such as a sample-evoked stimulus-specific response, could be retained during a delay period by short-term potentiation mechanisms, such as brief changes in the state of the synaptic weights in the network, such as the increase in residual calcium concentration in the presynaptic terminals. So the idea is that this information can be retained in the absence of active spiking activity in the synaptic weights in the network that code for this specific item. And then the model further suggests that if a non-specific burst of energy is propagated through the network, and here we're using transcranial magnetic stimulation, if it's applied to the network during this period, then that readout would appear structured due to that residual pattern of synaptic weights. Okay, so how do you actually measure this in humans to see if this is how it's actually happening? There's some evidence from this from neurocomputational modeling and recordings in petri dishes, but what about in humans? So how do we measure activity silent working memory? We address this first by having subjects perform a one-item delayed recognition task that requires the short-term retention of a direction of motion or a face or a word, and we trained a pattern classifier to identify areas of the brain important for retaining these categories during the delay. And then in a second phase, in which we simultaneously record the EEG, and we apply a TMS, a single pulse of TMS to areas identified in the fMRI task, subjects perform this two-item task with the retro cues, and we apply the TMS shortly after the retro cue comes and causes one of the items to be in an attended state and the other in an unattended state. And then we deliver a single pulse of TMS to one of the three areas involved in representing the three categories. And we record and decode the EEG, the TMS evoked response, to see how the TMS pulse propagates through the network from the stimulated site to structurally and functionally coupled regions at that point in time. Okay, now applying a burst of TMS while recording EEG is not without its challenges. This is what an ungated TMS pulse looks like on EEG. I don't know if you can see the scale there, but it's 600 microvolts. The Nextim TMS system that we used, the amplifier gates recording around the TMS pulse from 100 microseconds to 2 milliseconds. After applying a median filter of 20 milliseconds around the pulse and using ICA to reject components that clearly capture the artifact of the response, like this one, we can actually extract a reliable TMS evoked response, even in channels directly under the coil. Okay, and we've reviewed many of the issues with this technique in a couple of papers, and Nigel Rogash put together a toolbox to help the field use standard procedures. So check those out if you're interested. And this is what the spread of the TMS evoked response looks like when participants are remembering faces and the face target is stimulated, or motion or words. And with this TMS evoked response, we use machine learning to decode the category that's being actively or passively maintained or is absent on that trial from the EEG data. But we don't have bottles or shoes, but faces, motion, or words. And we spectrally transform the EEG data and calculate power and frequencies, 34 frequency bins between 2 and 50 hertz for each of 60 electrodes and each of 192 trials. And these are the features used in the machine learning analyses using a leave one trial out K-fold cross-validation approach to classify the presence or absence of each category on trials when that category is cued or uncued or is absent. And so decoding the EEG, we see a similar pattern where both items are initially decodable. This return to baseline for the uncued item. This is a classifier area under the curve now. That is the extent to which the classifier is sensitive to the differences between trials in which the category is present and either attended or unattended, and trials in which it's absent, which is an empirically derived baseline. This is essentially the proportion of trials that the classifier correctly identified whether the category was present or absent, averaged across the stimulus categories and the TMS stimulation sites. And when we applied a single pulse of TMS to a brain region, typically in posterior parietal areas that we think is involved in retaining this information in a silent but potentiated state, we can see a TMS-induced reactivation of the neural trace of this item. And that previous analysis collapses across stimulus category and stimulation site because this Frankenstein effect, as it's been called, did not interact with stimulus category. But the question here is, does the effect vary with the site of stimulation? This is decoding the presence of the unattended item for the TMS targeted category versus the non-targeted category. So words when we're stimulating the word site or versus the face or motion area. And the Frankenstein effect is larger for the type of stimulus that was targeted, but it still occurred for the non-targeted stimulus too, presumably because the site of stimulation, the evoked signal propagated to task relevant regions associated with that, the other categories. And okay, so we were excited by this finding initially, and we decided, okay, we need to replicate this to make sure it's real. So we extended it and made sure it was real. We also jittered the TMS onset to rule out any sort of anticipatory response. And we targeted the same region for all subjects now in the posterior parietal cortex, the precuneus, in an area that was suitable for TMS EEG. And this was the area that was the peak motion decoding region across all subjects. And then we also applied the pulse during the first delay when the unattended item was still relevant on the trial. And also on the second delay, after that second RetchEQ says, okay, now you can drop that uncued item. It's no longer relevant on the trial. And we replicated the decoding of both stimuli before the first RetchEQ. It's more smeared out here because we jittered the delivery of TMS and we're epicking the data to the TMS onset. And we replicated the Frankenstein effect after the first RetchEQ. But during the second delay period, when that uncued item was no longer relevant for the trial, we did not see the Frankenstein effect, presumably because the subject had actually dropped the item from working memory because it was no longer relevant. And this was also replicated by other groups a couple times now by Michael Wolf and Mark Stokes' group, and others have as well now. We also established an effect of TMS on behavior. So single pulse TMS didn't have any effect on participants' ability to remember the cued item, but we were only ever testing the cued item. That is, the cues were 100% valid. We wanted people to really believe the cues and shift their attention to the cued items. But if TMS was really reactivating the uncued item, and it was being brought back into the focus of attention, then people should have a harder time rejecting matching lure probes of the unattended item if we were testing their memory for the cued item. And that's what we found. So TMS induced more false alarms on the first probe than controls when the uncued item was still relevant on the trial. But TMS did not affect the second probe when it was presumably dropped from working memory because it was no longer relevant. And this too has also been replicated by Jackie Fulvio with some further control conditions as well. So here's the depiction of how this could occur according to the synaptic theory. So we could decode both items when they were presented and initially maintained in working memory. Following the first retrocue, the uncued item returned to baseline. And by that, I mean the same decoding technique that could decode it when it was being actively maintained could no longer decode it when attention was prioritizing the maintenance of the cued item. According to the synaptic theory, short-term synaptic plasticity mechanisms like the calcium concentration, the presynaptic terminals of the neurons in the network that represent that item could be coding for that. And because this activity silent item was still potentially needed on the trial, we think it was still in working memory, and TMS was able to reactivate it and it biased behavior. But when it was no longer relevant on the trial, TMS could no longer reactivate the item or affect behavior. And the main point I want to make is that so we can use TMS with neuroimaging kind of like analogous to sonar to ping the brain and detect otherwise unobservable representations. And this reactivation effect is consistent with the synaptic theory of working memory, and we were able to test it non-invasively in awake behaving humans. Now, there are a number of plausible neurobiological mechanisms that could support this, and it is not clear which one is correct yet. We haven't figured it out, and there's some encouraging results, some interesting results. Barbosa has some interesting the replication of this with a neurocomputational model seems to, the effect of TMS seems to have an important effect of reducing variability in the signal that is associated with this response. Nonetheless, so Mark Stokes has a nice review about the variety of mechanisms that could support this. It's not entirely clear which one is correct yet, but they're all short-term synaptic plasticity mechanisms. The hypothesized mechanism I want to focus on right now is this long-term memory account of latent working memory. And groups from University of Chicago and Princeton have suggested that latent working memories are activity silent because they're not in working memory. When subjects drop them, they are displaced to long-term memory, and so they're not represented by active maintenance mechanisms. They're supported and represented by long-term potentiation mechanisms, kind of like they're in the beginning stages of long-term memory consolidation. This is an important account to test, but it's an alternative account of the latent working memory. And so the idea is that they're displaced to long-term memory, and bringing it back into focal attention must involve episodic long-term memory retrieval processes, even though it's a few seconds. Like if you think back to the initial example with Savannah, when your attention shifts away, how do you get that name back? There are models that suggest that even though that's a delay of a few seconds, that really involves the same kinds of episodic long-term memory retrieval processes as episodic memory does. Foster and Ed Vogel and O have recently proposed that items in working memory, in order to consider them in working memory, they must be represented by neurally active signals. And when its neural representation is no longer active, it's displaced to long-term memory. So these are competing accounts for this phenomenon. So one is a short-term plasticity-based mechanism. The other is a long-term plasticity potentiation sort of account. So how do we test them and arbitrate between which one is correct? Well, the cognitive literature is informative here because accessing displaced working memory items by retrieving them from long-term memory, meaning using episodic long-term memory retrieval processes, involves retrieval practice that benefits your subsequent memory. So your ability to access the information on subsequent long-term memory tests should be enhanced. Much more so than continuously maintaining it and reporting it directly from focal attention. This is the so-called McCabe effect. And there's a really rich history of excellent work that was done on this topic in the 70s in the cognitive literature. And this has really resurfaced and grown since the late 2000s. So we can compare working memory performance and subsequent long-term memory of those items to help reveal the ways in which they were initially maintained on the working memory task. So in terms of a neurocognitive model, working memory representations appear to be incredibly distributed and dynamic. So from our view, it's technically not accurate to think about items existing in distinct working memory and long-term memory stores, sort of a traditional dual stores account. And as the academic son of Fergus Craik, I feel like I have to comment on this and advocate for this processing-based approach. And in some reviews we've received recently, there seems to be a suggestion that there's a difference between working memory encoding that's distinct from long-term memory encoding, in the sense that when attention switches away from an item in working memory, you can somehow encode it or re-encode it into long-term memory. This seems to be the way some computational modelers talk about it. But in our view, as soon as an item is a stimulus is perceived, that's when encoding happens, and there will be a trace that is represented and consolidated in memory, regardless of whether one is performing a working memory task or a long-term memory task or no memory task at all. In Craik's words, encoding activities presumably happen at the time of initial perception and learning, and such representations can be actively maintained during working memory tasks. That's what people typically do. And then they can be subsequently accessed and influence performance on long-term memory tests. We argue in these papers that there's no such thing as working memory and long-term memory encoding, as reflected by that suggestion that after a stimulus is presented and processed, like upon presentation of the retro cue, that a participant could somehow re-encode it, the uncued item, into long-term memory instead of working memory. So that distinction doesn't really make sense. Again, working memory, long-term memory describe classes of tasks or situations, and many different types of processing can be involved in either class, some similar, some different. Sure, when people expect a long-term memory test, they tend to engage in deeper, conceptual, more elaborate processing, which typically results in memory representation being more accessible on subsequent long-term memory tests. But this should not be taken to mean that when people drop an item from working memory maintenance, such items are encoded into long-term memory instead of in working memory. Okay, I'll get off my soapbox about that issue. One of the ways in which we are addressing questions about the roles of attention and long-term memory and working memory in my lab is by examining the nature and source of age differences in working memory between healthy young adults and older adults, including those with varying degrees of risk for Alzheimer's disease and related dementia. Age differences in working memory may be large, small, or non-existent across different situations, and this seems to depend on the extent to which the tasks really tax age-related deficiencies in either attention control processes or episodic long-term memory processes. This two-item double retrocue task that we've been working with looking at subsequent long-term memory of the items is particularly informative for assessing the relative contributions of these constructs. And we think we have a pretty good handle on what causes age differences to be large, small, or non-existent in these types of tasks. And we're about to submit our dual mechanisms of cognitive control account of age differences in working memory. And so I encourage you to check that out if you're interested. I'm not going to talk about it too much because I think folks will be more interested in the work that my postdoc's doing, decoding the activation of working memory items from ERPs during the delay period and stimulus evoked response of an NBAC task in healthy older adults with various Alzheimer's disease biomarkers, which I'll describe in a moment. So I'll just give you a summary of our tests of this long-term memory account by looking at subsequent long-term memory of the items from this working memory task with two items and the attention switching situation. I'll just summarize these by telling you that there were three factors that had significant effects on working memory. So working memory was worse for items retained outside focal attention. It was worse for the older adults than younger adults, and worse for younger adults with divided attention. So these three factors all affected working memory performance, but none of them had any systematic or interactions with subsequent long-term memory effects. So this dissociation is sort of consistent with, or is not consistent with that long-term memory account of latent working memory. So whatever maintenance process is involved to help retain items on this working memory task when you're switching between items, it appears to be distinct from episodic long-term memory retrieval. So again, I think these results are inconsistent with the long-term memory account, and we think they're better accounted by the synaptic theory and a dynamic model of working memory. Now, that's not to say that long-term memory is not involved in working memory performance at all, ever. I mean, there are plenty of examples like the McCabe effect that I just mentioned, as well as hippocampal amnesics, working memory deficits and hippocampal amnesics. There are plenty of examples, as well as neuroimaging evidence of hippocampal activity during working memory performance. And as I said in this paper, when episodic long-term memory seems to be involved in working memory, it appears to be really dynamic and highly context dependent. So understanding these sort of dynamics is a really interesting, important area for the field going forward. Okay, so now getting back to this idea about older adults with Alzheimer's disease biomarkers, these people who are or will have long-term memory deficiencies. How is their working memory performance? How is their ability to maintain information in working memory looking? So the question is whether latent working memory retention is associated with factors that negatively affect long-term memory. If these activity-silent working memories are in the beginning stages of long-term memory consolidation, then this delay period representation may be sensitive to factors associated with Alzheimer's disease. We don't have an Alzheimer's disease research center or even a medical school at Notre Dame to get access to well-characterized patients, but we do have this great ongoing longitudinal study of older adults, and we were able to measure cognitive functioning participants in the study, and we recruited, scanned, and tested 50 of the MRI and TMS eligible participants. And so far, we have data to report for 4 biomarkers for the current study: age, cognitive function from a single time point, whether they have 0 or at least 1 copy of the ApoE4 allele, which is the strongest genetic predictor of AD conversion, and the personalized AD cortical thinning signature from structural MRI from a local clinic on their 1.5 Tesla scanner. So this personalized Alzheimer's disease cortical thinning signature from Brad Dickerson and colleagues has shown that Alzheimer's disease is reliably associated with this signature of cortical thinning in these specific regions relative to normal aging. So we're using this PADI measure as a biomarker of cortical thinning from a single time point, and we'll be looking at changes in this longitudinally over time. In terms of ApoE4 status, cognitively healthy carriers often do show deficits on long-term memory tests compared to non-carriers. And with regards to working memory, one fMRI study has shown that carriers don't have cognitive deficits on working memory, but they do show increased brain activity during a one-back test compared to controls. And so this suggests that even though their performance is intact, the way that their brains are performing the task is different. And it was in the sense that more activation was required in order to perform at that similar level. Perhaps due to some deficiencies with emerging processes that support either short-term synaptic plasticity or associative bonding or these long-term potentiation mechanisms. But this is fMRI, so it's not clear if this relates to deficiencies during encoding, during the maintenance process itself, or during the recognition retrieval phase. So it would be much better to do this with EEG or MEG. And this is just the bulk univariate analysis, so it would also be better to do this with decoding procedures to look at the nature of the representations that are retained. And surprisingly, only one published study has attempted to decode stimulus category from a one-back ERPs, and this was only recently published in healthy young adults. And so this seems like some low-hanging fruit for any eager grad students or postdocs, especially since there are tons of open access NBAC data sets out there, neuroimaging data sets, where there seems to be a lot more work that should be done decoding stimulus representations during NBAC tasks in young adults and older adults. In this study, they found that object versus scene stimuli could be significantly decoded during stimulus and response periods of the trials, and also approximately 2 to 5% better than chance, so not great, between 860 to 1275 milliseconds, so a brief period in the delay period. Certainly not a sustained elevated decoding throughout the delay period. And the authors didn't report any association between decoding accuracy and working memory performance. And they also tested and recorded EEG during a 2-back version of the task, but they didn't report any decoding accuracy. I have some ideas as to why, and I might touch on that later. In our study, participants performed a 1-back task with scene, object, and face stimuli while we recorded EEG. And we're most interested in this delay period decoding, whether delay period decoding shows that the item is actively maintained or passively retained. And so we're looking at after the stimulus is presented, when you're retaining a scene, this delay period here, and then during this period when the probe stimulus is presented, and it's either a category repeat or a non-repeat. And if we can decode the category of the stimulus that's being processed, but we're really interested in this delay period. And there are some different accounts and different predictions that we made depending on whether the groups with Alzheimer's disease biomarkers have intact or deficient performance relative to the biomarker negative group. And I'll just skip the predictions if there are deficient performance, because on the 1-back task, there were no deficiencies with any of the biomarkers groups. So I'll just focus on the predictions for when performance is intact. If there's active maintenance and the biomarker plays a role in working memory performance, then we predict a difference in the level of decoding representation for the biomarker plus versus minus group. But if there's passive maintenance, like the activity silent account, if the biomarker doesn't play a role, that is, these biomarkers associated with emerging deficiencies in long-term memory is not playing a role, then we shouldn't see any difference in the delay period representation. But that long-term memory account of latent working memory, if the biomarker associated with long-term memory deficiencies is playing a role, then we think that there should be a difference between these groups during that delay period. And we predicted that it might be in the form of the groups with deficiencies in long-term memory. If they're not able to rely on long-term memory, then they may be forced to engage in more active maintenance during the delay period. So consistent with that fMRI study that was shown elevated signal for ApoE4 characters, but specifically during the delay period, which they were not able to distinguish. Okay, so those are the predictions. Relatively standard EEG and ERP preprocessing and decoding procedures, and performance was intact for the group that had more versus less of a cortical thinning signature. Again, these are healthy older adults who do not have much deficiencies, except on the 2-back task. The groups with it was a continuous measure. So people who have more of a signature of cortical thinning performed worse on the 2-back task, but not the 1-back task. This was associated with the neurocognitive function, fluid crystallizing, and executive functioning. Okay, here's the decoding performance. This is separated for those category repeat trials. So we're decoding face. This is an example of a face decoder. After that face is done being presented, the stimuli, the intertrial intervals jittered. So we're looking at just the items that are purely after stimulus presentation is being done. So this is just delay period representation. There's a lingering trace of active maintenance for the group with more evidence of cortical thinning than the low PADI group. Certainly not evidence of active maintenance during the delay period. We have nice strong decoding in the stimulus evoked response, especially when it's the same category that's being repeated. Interestingly, this is decoding the face stimuli, the grayed out version. This is decoding the face stimuli when a scene or an object is presented. And so that we actually get pretty strong decoding of face information when the object or scene is being presented, because they're comparing the stimulus that they're processing to the item that's being maintained in working memory. So this is like a retrieval related representation. No differences between those groups there. There may be some slight association during the delay period for people with this more evidence of cortical thinning signature that's really specific to Alzheimer's disease relative to normal aging cortical thinning. And we also did a multiple regression analysis to look at a variety of the biomarkers, the role of the variety of the biomarkers during the delay period, the stimulus evoked response, and accuracy and RT. And the PADI has some predictive utility in predicting the delay period representation, whereas none of the others do yet. Again, these are all healthy older adults in a longitudinal study. And so if we can get some funding to test more subjects and to test them repeatedly, we'll be looking at this longitudinally over time. And we'll be able to hopefully be able to predict who is developing the greatest risk. Okay, so in summary, healthy older adults with versus without the biomarkers have intact behavioral performance on a one-back task, really easy task. But we were able to pick up some differences in the way that the brains were retaining the stimuli during the delay period, not during the stimulus response, stimulus evoked response, or that recognition retrieval comparison process. And we still have a lot of work to do on these data. We're going to try to decode the frequency transform data. We'll try to decode the actively and passively retained items on the two-back task. At first, I couldn't believe that there aren't more papers that have done this. This seems like such low-hanging fruit. But when we tried to code the labels for a continuous end-back task, which item is in the passive versus active state, it's a lot trickier than I initially thought. But we think that we figured it out. So again, I think this is low-hanging fruit, especially with a lot of open access data sets out there. So if anybody is interested in trying it, please get in touch with me. I think we've worked out a scheme for coding the labels according to passive or active states, or absent or relevant states. And I think that's why the Ratcliffe paper didn't, they recorded the two-back data, but didn't report any decoding analyses, probably because it didn't work, because it's really tricky. But anyway, I think we've figured out a way to do that. And then we also have some other biomarkers to examine, especially these blood-based measures of amyloid and tau deposition. And we'll be looking at longitudinal change in these biomarkers. So we'll have multiple time points of subjects and these other biomarkers if we can get some funding to extend this and expand upon it. Okay, so I am going to transition to part B now. And this is really kind of a short phase. So I could kind of take questions on part A now or wait until the end.
Let's wait until the end.
Okay, we'll wait until the end. I'll just try to touch on this really quickly, how we can use TMS to modulate long-term memory with this systematic review and meta-analysis, and provide an example. Okay, so we really want to see how TMS can be used to enhance episodic memory, especially in older adults with Alzheimer's disease biomarkers, to help preserve and enhance memory functioning, ideally with transfer to real-world behavior. But there are so many intrinsic factors like these in blue and extrinsic factor like these in light orange that can impact the effects of TMS. Obviously, we know how TMS is administered is important, where you're stimulating, how you determine the stimulation site, the type of control conditions that are used for comparison. And one needs to be careful when making design decisions based on what is known about TMS in motor cortex and then applying it to other regions of the brain, because there are some complex and surprising effects that can have unintended consequences. Like vertex stimulation is really common control condition for comparison, but there's some evidence that TMS to vertex can actually activate default mode network processing. So that's a potential confound when you're trying to compare. We know that default mode processing is really important for our episodic memory functioning, and so that can be a difficult confound when trying to make comparisons between an active stimulation and a control site when you're stimulating vertex. And then there's also aspects of the behavioral experiment, such as the type of stimuli, the cognitive process that's stimulated, and the type of assessment. So my student, Nick Ye, and I conducted a systematic review and meta-analysis of studies that have used TMS and measured episodic effects on episodic memory. We tried to get all of them and then code them for important characteristics. So here's the full table. You can't even see that without squinting. I'll zoom in, you still can't even see it. So I'll just show the first couple of rows to point out that we coded for characteristics, whether what type of TMS intensity, whether it was online, offline, the phase, whether it was during encoding or retrieval, the targeting procedure, target area, control site outcomes, and what the results were. And Nick extracted as many effect sizes as he could, both between conditions within a study as well as between studies. So this is a multi-level meta-analysis to examine potential moderators and two-way interactions, because interpreting the overall aggregate effect is complicated by the fact that a lot of studies are designed to induce impairments to assay the causal role of a stimulated brain region in the memory performance. Or enhancements. And so if you just look at the net aggregate effect, it is likely to be null because of the goals of the study. And so there's a lot of interactions. And some of the key results that were generally larger enhancing effects of 1 Hertz RTMS relative to higher frequencies, which is kind of surprising if you compare that to what is known in the motor literature. Especially when applied below motor threshold intensity. So there are some interesting effects of sort of near threshold or just slightly below threshold stimulation. And there was an interaction between the effects of 20 Hertz RTMS, where there were generally impairments when applied online, when you're doing the task, versus offline, where there were improvements. And surprisingly, in aggregate, there were not reliable differences or interactions with stimulation site, whether it was applied to the left or the right hemisphere, to frontal or parietal cortex, or whether it was applied to impact either encoding or retrieval processes, or whether the outcome assessment tested more recollection versus familiarity-based assessments of retrieval. And there also were not reliable aggregate differences between targeting procedure control conditions even. Now, there's some caveats here. I don't want the interpretation of this to be like, okay, great, we don't have to worry about having good control conditions or a stimulation site, because there are definitely considerable differences in the strength of the inferences that you can make about the results based on these methods. And also, this is for accuracy-based measures only. So we didn't consider reaction time or the subjective experience of remembering, like confidence judgments. And the narrative review revealed some interesting effects of parietal stimulation on the subjective experience of remembering. The main caveat is just there's not enough data points in all of the cells to be able to reliably analyze all of the combinations and permutations of parameters that are likely to interact here. We highlighted areas where there were insufficient data for valid inferences so that we could try to encourage the field to fill in these gaps. We also published the table as an open access data set. So the goal was so that the database of RTMS effects on episodic memory could be added to facilitate future meta-analysis. So please check that out and contribute. With regards to older adults and clinical populations, there really were not enough studies published to provide valid conclusions based on these effects. There's a qualitative comparison about the effects revealed on older adults with some clinical conditions. So enhancing effects of 1 Hertz stimulation and impairments of 20 Hertz stimulation, online stimulation, and enhancing effects of 20 Hertz offline stimulation that have been found in both older adults and patients with MCI or Alzheimer's disease. And finally, just quickly, I want to show an example of how we used RTMS to enhance memory on an emotional memory trade-off paradigm. So during coding, participants viewed a valence image, so a car crash on a neutral scene, and then provided a valence approach avoid scale, a rating for this, and then received 2 seconds of theta burst stimulation after every block of 8 images for 16 blocks. This was done to either the dorsal medial prefrontal region that is functionally connected to the ventral medial prefrontal region that was associated with the selective preservation of negative objects in memory from an fMRI study, Payne and Kinzinger. And we targeted the dorsal medial prefrontal region under the assumption that theta burst stimulation would modulate neural activity in excitatory manner, similar to intermittent theta bursts. These were brief trains, 2-second trains of the same protocol as ITBS to both the target site and its associated network. So we think that stimulating here will propagate to the ventral medial region that we know is identified in playing a role in this emotional memory trade-off effect. And then we had subjects perform a recognition test to assay their memory for negative and neutral objects separately from the neutral background scenes. And what is typically seen in this emotional memory trade-off effect is that people are better at remembering negative objects, and that occurs at the expense of poorer memory for the background scenes. This has been described as analogous to the weapons focus effect. Like if you are robbed, you tend to focus on the weapon and remember the weapon really well, and then you lose details of the background, like the perpetrator's face. And that's relative to neutral information. And the finding was that the ITBS2 medial prefrontal regions has an enhancement effect of this emotional memory trade-off effect for negative objects relative to the scenes. Only at the long delay, this emotional memory trade-off effect tends to be exaggerated or really associated with following a night of sleep. So this is with a long delay, 24 hours, whereas there's no effect on a short delay. So really sort of context dependent. And so this is an example of how RTMS to medial prefrontal cortex can causally enhance episodic memory in healthy young adults. And at least one study has also shown that continuous theta burst can eliminate the perseveration of these negative memories in healthy young adults. And so this has clear therapeutic implications for disorders like post-traumatic stress disorder and other diseases associated with perseveration of negative memories. Okay, so I'll stop there and just summarize that RTMS can have powerful modulation of memory in both healthy and diseased adults, but there are really complex interactions among many factors that can be manipulated. And single pulse TMS with simultaneous neuroimaging can really provide a potent way to measure the state of memory representations and processes. And I hope I've convinced you of some of this. With that, I want to thank my students and collaborators who helped conduct this research, and the NIH and NSF for supporting it. And I'm happy to take some questions to clarify anything.
Thank you very much. So I just want to remind our audience, if you have any questions for the virtual audience, please just write in the Q&A box or raise your hands. And here as well, please, if you have any questions, raise your hands. While you're preparing questions, I actually have one question. It will be a bit controversial, I believe. So when in the science paper, you are actually just telling people, and people do know that they might actually have to remember this second unattended item afterwards. So that's actually, so they don't completely drop the item from their memory. So it's not maintenance. What do you think it is that you see the effect? I don't think that it should be completely maintenance, but maybe attention.
Maintenance of the unattended item?
Yeah, because I mean, you do. I mean, if it is completely irrelevant, you just cannot. You don't see that effect at all.
Yeah, so this is what you see with single retro-Q studies, where especially when the retro-Q is 100% valid, where you're never tested on the uncued item. So right after that retro-Q, you can drop the item.
Yeah.
The two retro-Q task is different in that subjects know that they have to maintain both items. After the first retro-Q, the uncued item is tested 50% of the time.
Yeah.
And that's always the case. They learn that, we tell them that, so they know that they can't drop that uncued item initially. Only after the second retro-Q can they drop it completely. Yeah. And so we think that it is maintained, even though we have this evidence that it's not elevated if this activity is silent.
So it is actually, it is activity silent. So it's actually
at least for the first half of the trial, which is when we ping it and can reactivate it. And then at the second half of the trial, the second retro-Q says they know they can drop it, and there we can't reactivate it and it doesn't affect behavior. So we thought that was pretty consistent with the synaptic theory. One important difference is that synaptic theory, suggests that, the calcium concentration has this long delay constant, so it can retain latent working memories for a really long period, like up to a second. This is like tens of seconds later.
Yeah.
And with this phenomenon where you can reactivate it when it's still relevant, but not when it is no longer relevant, that suggests that this is under cognitive control.
Yeah.
So there's some mechanism that can keep this while it's still relevant and then clears it so that, and there's this active deletion mechanism from the cognitive literature that's discussed. And this could be an important function of working memory to clear the contents of working memory so that irrelevant information does not interfere with your ongoing cognitive functioning. So we think it was consistent with that. There's obviously a lot more work that needs to be done to follow up on it. So
Okay. Yeah, but I mean, if that's the case, you are also maintaining the attended item. So, but you can also decode from first. So, are we decoding attention instead of maintenance there? You think?
On situations without that double retro cue, yes, we argue that in the majority of working memory studies, working memory retention is almost perfectly confounded with attention, because the items that you have to retain in working memory are the ones that you have to attend to and are tested on. Only in the situation with the two retro cues do you get the separation between, here's this item that you are really prioritizing, you have to hold on to this one, that's the item you're going to be tested on. This other one, you can't drop it. And so this is where it's interesting, because some models suggest that it's only two items. The capacity of working memory, according to some models, is at least 3 plus or minus 1 chunk. So why are we showing activity silent of this item when you should be able to maintain both? But we don't.
Maria?
Thank you very much for the info. I have actually a kind of a question. What are you saying that, same idea of this or SNF theory may be applied for the system, because we have, like, when we have a situation when you can, there are lots of tasks when you have to get somewhere, and you have, let's say, 2 targets, and you will be able to choose between these targets only after you start it, and we know that this is something which is actually going on inside all the time, because that time, like, just based on the muscular, contraction based on the EEG is really short. So I wonder, like, maybe, the same principle probably be applied for the motor system, and, like, in connection with this question, I didn't really get what were the areas where you stimulated, how much they were different among the participants, and if you, as far as I understand, you did it once, but, like, if you do it twice, whether these areas will be reliable, like, how much is there a cross-subject variability for these areas, and intra-subject variability for these areas, what would you expect? And again, because if you speak about the motor, I wonder what might be the area we want to stimulate.
Great questions there, Maria. Yeah, you hit on a couple really important points.
Could you just repeat the questions?
Yes. So let me take the variability.
No, wait. Not because of that, because the Zoom people can't hear.
I'm going to repeat it perfectly. No, my working memory is horrible. That's why I study memory. But I do get the point. And so I'll start with the second one about the location of stimulation. So in the first experiment, we personalized. So we did the decoding procedure for each subject with a whole brain searchlight analysis, and we stimulated the sites for each category that were relatively distinct for each subject in similar regions, mostly in posterior parietal regions, but there was variability. Yet we saw similar kinds of effects stimulating different categories. And so in the second experiment, we showed this, that yes, the reactivation effect was larger when we were stimulating the targeted area, and that was the category of the unattended item. But we also showed significant reactivation when we were stimulating the non-targeted area as well. And so that was a little puzzling, that it seems to be more general. perhaps because of where we were stimulating in sort of regions of the parietal cortex, which is well, they were defined by the peak decoding accuracy. So it was variable, intraparietal sulcus, posterior parietal, like precuneus. The TMS intensity was it was based on the E field estimates from NextStim, and I forget exactly the volts per meter that were estimated to be induced. It's in the paper. Right, because the sites and the depth of the stimulation were slightly variable across subjects, we matched the E field for all subjects. So the intensities would be slightly variable. That actually didn't have any correlation with the size of the reactivation effect. That was, yeah, that's a good question as well. And then in the second experiment, we chose the same site for all subjects, and stimulating the peak motion decoding site was also effective at reactivating words or faces. So that could be because this is a sort of more of a domain general attentional control network involved in the frontal parietal circuit. So the hypothesis was that stimulating here might cause the propagation of signals to the domain relevant sensory cortex. We analyzed the source localized signal. We got some sort of consistent evidence, but it wasn't really clear or strong evidence. So that's in the supplemental materials. And then subsequently, we've been trying to follow up on this by stimulating sensory cortex and presenting the stimuli at the site where we're stimulating in visual cortex. We've been having trouble getting really clear, consistent results of that. So I haven't published yet. I haven't felt confident enough to publish on that. But actually, I think Yurko has some of the strongest evidence consistent with this sensory recruitment hypothesis of the impulse sort of reactivating, it's spreading to sensory cortex and reactivating the items. So that was the first or the second question. Oh, and I'm showing my working memory. My attention shifted. Now I need to.
The first question was how much same principle might be applied for more than this.
Yes. Yeah. Okay. Yeah. Yes. So this relates to one of these alternative accounts of activity silent representation. And so maybe the reason we can't decode that item is because you're not maintaining the sensory representation. It's a transformation of that item. And in many situations, say you can remember, you have to, and there's evidence for this with delayed saccade task and neural recordings in frontal eye fields in monkeys performing this, where, and this is one of the reasons that sort of led to this dorsolateral prefrontal cortex is like where working memory is stored, because you see these cells that show elevated, sustained, or that's actually debatable. Some of them have more of a ramping up, some cells have more of a ramp down, but that's in the frontal eye fields. So those cells are coding for the eye movements that are required to move the eyes to the location where the stimulus is presented so that you can get the squirt of juice and get your reward. That's not in visual cortex. The retinotopic location of what spatial location was presented and retained in working memory doesn't show that same kind of elevated signal. But you don't need that if your response to get the reward is a motor action. So an account of activity silent, one of them is that the representations are transformed to like a prospective action plan. And so I think the motor system definitely plays an important role. Like what type of motor response is required on the working memory task is certainly likely to play a very important role. And those are things that could be decoded and tracked. Like if you have to perform a left button response versus a right button response with something like the lateralized readiness potential with EEG. Great question.
Any other questions from the audience? Yes.
Do you have any best guesses for the biology that would allow the unattended items to persist? And you say it's not complicated.
Yeah, best guess. That transformation, and there's some evidence for that and actually, so some evidence that I have for that comes from phonological short-term memory, which appears to operate differently than visual short-term memory. So, and I think this makes sense if you think about the differences between how the brain retains visual information versus temporally unfolding auditory information. So if you need to reflect back on a visual image, you can just move your eyes back to the environment and typically refresh. So you need like a retinotopic representation of what that is. Auditory information unfolds and it's gone. So there it makes more sense for some sort of a buffer type system to hold the sort of temporal sequence of auditory stimuli so that you can bind them together and integrate them with preceding and succeeding information to establish more complex representations. And so we did a phonological short-term memory fMRI study with a similar type of procedure, training a classifier on auditory cortex and while people were perceiving speech versus non-speech stimuli, and then retaining them in phonological short-term memory. And we did not see a similar kind of a pattern where the sensory representation that was initially established appeared to shift. So it was like a transformed representation. So there wasn't delay period decoding in auditory cortex, but there was in like the phonological buffer around supramarginal gyrus, for example. But if you train the classifier during the delay period, then you could decode speech from non-speech stimulus. So it's like in auditory sensory cortex, there was the information that was being retained, but it was in a different format. The pattern was different than the pattern that was elicited during encoding per se. So I think that this transformation account, so it's not that there's no neural activity at all. I mean, even spontaneous activity in those regions will be occurring, but that spontaneous activity, let's say you're like bubbling spontaneous activity, there would be like a little reactivation every time from spontaneous activity, but that probably is not detectable from scalp level EEG or fMRI. So you could probably detect it from spontaneous activity if you have really, really high resolution recording, like in intracranial EEG, for example. And this temporal or this transformation account is probably most common. But the long-term memory account is certainly plausible as well. We just have not found, we tried to test it, we tried to provide evidence for it in a number of different ways, and we've not provided much support for that account.
Any other questions? Okay, I had just one very short, then we will stop actually there. For long-term memory account, actually, isn't it a bit too when you think of it from a like I don't know, energy conservation point of view, isn't it a bit too disadvantageous to actually put something that you think that you want to keep into long-term memory storage? And I mean,
yes, that's a really good question as well. And usually the way it's framed is maintaining information, every potential representation that could be goal relevant in an active manner, having spiking activity. Action potentials are very metabolically expensive. So if you were to go around the world maintaining every potentially relevant representation via spiking, that would be a really inefficient way of doing it. And evolution and the brain is not inefficient. One thing I heard once was the brain, according to the principle of thermodynamics, the brain would literally boil in seconds if that's how it worked. So this activity silent mechanism, these short-term synaptic plasticities, is a much more metabolically efficient way to do it. But does that mean that every potentially relevant stimulus you encode when you go throughout the day is a long-term memory is formed? Probably not, because that would really muck up your long-term memory. But does it mean that the beginning, the very earliest stages of long-term potentiation is occurring, these really initial consolidation? Possibly. And there are many different forms. There are at least 6 different forms of long-term potentiation mechanisms. So it could be the case that every potentially relevant stimulation, an initial cascade of processing that starts memory formation is occurring. There isn't enough time for new dendritic sprouts to happen. There's not enough time for protein synthesis to occur. So it could be these really short-term, there's actually like short, long-term potentiation mechanisms that could start the process. But if you then decide that that is no longer relevant, these representations could be cleared. So, sleep, for example, one of the most important functions of sleep appears to be a dendritic pruning of all the information, these memories that you start to form over the course of the day that are just mundane, not relevant, they get cleared with time. And this might be like the very earliest stage of those consolidation processes, which is why I'm really interested in applying this to aging and Alzheimer's disease, because I think this might be like a really fundamental source of the beginning stages of memory deficiencies that have not really been well characterized. So I think this could reveal a really important target for memory-related disorders.
Yeah. Thank you very much. Thank you to our audience, and thank you very much for this very interesting talk. See you next week.
Thank you.